Alright, in this video I would like to talk about some of the applications of logarithms related to both data and also models. And so I want to start by just thinking about what the logarithmic scale or the logarithmic number line allows us to do. So if I think about the usual number line, numbers are spaced according to their distance. So for example, the number 1 and the number 5, they're spaced 4 units apart, and any other numbers that are 4 apart have the same gap. So for example, 1001 and 1005 are going to have the exact same space between them, even though the space between 1 and 5 and 1005 is huge relative to that size of 4. A logarithmic number line, first of all, only has positive numbers because we're taking powers of a base, and they're spaced according to their multiplicative distance. And so what I mean by that is if I thought about the space between 1 and 3, that space of 3, that's the same as the space between any other two numbers who are multiplied 3 apart. So for example, 3 times 3 is 9, and so the space between 3 and 9, let me mark that. The space between 3 and 9 is the same as the space between 1 and 3. So this distance is identical. Similarly, if I go from 10 to 30, that's a multiplicative scale of 3 as well, and so this is also that exact same distance. So a logarithmic number line allows us to think about how far different numbers are apart in a multiplicative sense. So that's the idea of the logarithmic scale. And now I want to share an example of something that I've done a little bit of research in with a biologist, and that has to do with abundance of transposable elements. So to give you an idea of what we're talking about, I have on the left a picture. It's a several cells, and we can see that their nuclei have been stained, so they show up in this different color. And what's significant about a nucleus is that inside of the nucleus, that's where all of our DNA is. And so here on the left, the furthest cell, we can see that the chromosomes are bright and lit. And so these chromosomes have all been, they're in a state where they're really compact, whereas in the other nucleus the DNA has been spread out throughout the nucleus. It's a different stage of the cell cycle. And so when scientists were looking at DNA, in this compact state, they were able to find patterns. So there were bands that would show up, and this is fairly famous understanding of chromosomes, that there are these locations that are different colors, they're different density. So we could identify patterns in structure, and these markers help us look at relatedness. Well, DNA that's in the chromosome is actually a really long polymer chain. So we have nucleic acids that's double-stranded. This is a picture showing how DNA is replicated, but we have this double-stranded helix of nucleic acids, and it's really long if you were to stretch it out. What I was interested in and looking at were if you unraveled that chain of DNA. So here, imagine this is one long strand of DNA, and we start to look at patterns of where the nucleic acids formed. There are certain sequences of those acids, of the nucleotides, that happen over and over and over again. So in this picture I have two color little markers. So this is at the molecular level instead of the band level. But we see that, so for example, these yellow marks repeat many times throughout the DNA. Similarly, we have some red markers also occurring throughout the DNA. And so there are a lot of these repetitive elements, and some of them are called transposable elements. What they do is, in the process of DNA replication, they copy themselves and insert multiple places in the DNA so that they, in essence, reproduce and have really high abundance levels. So I was working with a biologist where they sequenced the DNA and found these patterns, and we were looking at how frequently different classes of these elements appeared. So this is not real data. I can't share that in this particular video, but the order of magnitude is the basic idea. So I have 5 sample transposable elements, and I'll name them A, B, C, D, and E. And so we have 2 different samples. And in sample 1, I have a particular abundance. So when we did the sequencing, we counted how many times we found an element that matched type A, and we found about 170,000 copies. And in another sample, when we looked for the same pattern, we found 83,000. So I did that for each of many different families, and here I only have 5. But what I'd like to highlight is the order of magnitude is really different. So class E had a very small, it's still a lot, 500 copies, but that's much, much smaller than A and C, which are in the hundreds of thousands of copies. So that's so order of magnitude says how many multiples of 10. So this is order of magnitude of 100, and here we have order of magnitude of hundreds of thousands. So that's a 3 orders of magnitude difference, a 1,000-fold difference in the scale. If I look at sample 2, again I have one in the scale of hundreds of thousands, and another here in class E that was only 80, so the scale of 10. If I were to plot the abundance levels, so here the x-axis is measuring numbers of copies, so I have from none to 300,000, so this is this data point matches the 295,000 for family C for sample 1. Notice that if I use the ordinary line, because we're spacing according to actual distance, we have a couple data points that have a similar order of magnitude, but all of the rest are clustered down close to 0 because they're smaller order of magnitude. It's even worse for sample 2. I have families A and C that are either tens of thousands or hundreds of thousands, and the other 3, even though their scale is 1,080, they're all clustered close to 0. And so when I look at data that have orders of magnitude difference, if I look at it in the normal number line sense, I'm going to end up having some numbers clustered close together, apparently, even though they're still orders of magnitude different. So what I do is I plot the same data, but on a logarithmic scale. So here I've got my x-axis. Instead of using a linear scale where numbers are spaced according to their distance, now they're spaced according to their order of magnitude. So notice now that the numbers 100 to 1,000, that's multiple of 10, to 10,000, that's another multiple 10, to 100,000. These distances between the new orders of magnitude is always the same. That's what the logarithmic scale is trying to do. And we can see now that my data, I can get a sense of where all of the data are, right? So that sample 2, class E, we can see that it's down here on the order of magnitude of close to 100. It's at 80. And at the same time, I can see these that are up near hundreds of thousands on the same plot, and everything in my graph has been spread out nicely. One of the reasons that we look at this data is I want to see how these samples are related to one another. And so what I want to do is I want to do a scatter plot where I take the same families and I say what's the abundance for sample 1, and compare it to the abundance of sample 2. And so we can see that if I do the linear scales, my orders of magnitude in the hundreds of thousands stand out here, and all of the rest of the points are clustered down close to the origin because order of magnitude wise they're much smaller. So this is really, in a linear plot, it's really hard to make sense of those data. On the other hand, if I use both axes on a logarithmic scale, so for sample 1 it's logarithmic and sample 2 is logarithmic, those data spread out and I can start to see a pattern. And one of the things that might appear, and this is what we'll be talking about next, is the data on the logarithmic scale, when they're spread out and we're just looking at order of magnitude, it looks essentially linear. And that I want to now start to think about how can I model data when it looks linear on these logarithmic scales. And it turns out that we'll see that allows us to model it with a power function.